Good morning, friends of plus 2. In previous topic, you studied about chromosome map and crisscross inheritance and crossing over crisscross inheritance. You repeat it and revise it. Today, we will study very interesting genetic variations. Previously, we have studied variations. And the variations are of two types: somatic variations and germinal variation. In germinal variation, the variation which are called by change in genes and chromosomes. Today we will study about the causes of the genetic variations. First cause, recombinations. And recombinations are by independent assortment and meiosis during gamete formation, crossing over, and reciprocal recombination, independent assortment, etc. And these are called recombination every time. And second is mutations. By mutation, there is sudden change in chromosome, and sudden change in chromosomes are called sports. And sports are permanent inheritable changes. Genetic variations are all inheritable, and these are sometimes lethal, sometimes these are useful. And the genetic variation, the mutations are of two types: gene mutations and chromosome mutation. Gene mutations are the change in the part of the gene, and the chromosome mutations are the change in part of the chromosome. I will show you by diagrams. For example, in the book you can see this type of chromosomes, left hand side and right hand side. From the book I will give you diagram. I will show you the diagram. In the book it is very good written. Suppose in this diagram of the book, number 1, the reasons of the chromosome mutations are deletion. Left hand side the genes are full, and on right hand side C gene is deleted, and the deletion of the gene causes variations in chromosome. Second reason is duplication, ABCD, EFGH, and sometimes AB is repeated. That genes are repeated, that is called duplication. In C case, inversion. The arrangement of the genes are inverted when reversed, and the reversing of the genes and DNA causes variations in chromosomes, genetic variations. And next is reciprocal translation. Reciprocal translation means just crossing over. The two chromatids exchange their genes, that is called reciprocal crossing over. After showing you reciprocal inversion, reciprocal translocation, exchange of genes by crossing over, the genetic variations are caused by recombination mutations, gene mutations, chromosomal mutation, and chromosomal mutation methods are deletion of the gene, duplication of the gene, translocation of the gene from one chromosome to other, inversion of the chromosome. It is purely crossing over. And this then second change is numerical change in chromosome. Numerical change happens when during crossing over, the crossover jointed chromosome do not separate. Non-disjunction. By non-disjunction, polyploidy is there. I repeat, when crossing over there, the half the gene chromosome, half the chromosome pass on one at one pole and half the chromosome at one other pole during first meiosis. The chromosome number of become half. If the chromosome number do not half, one or two gene they don't separate by crossing over. Both the gene pass over in one pole. So two gamete will be formed. They will be defected gamete. Defected gamete, I mean one gamete will have 24 chromosomes and other gamete will have 22 chromosomes. And 23, instead of 23 it will have 24, and other gamete will have 22, one less. This is called polyploidy. You understand? That is called numerical polyploidy. In plants it is diploid, triploid, tetraploid. But in human beings there is genetic disorder by numerical changes of the genes or chromosome. Homologous chromosomes do not separate and pass in the one gamete, and they has 24 chromosomes, and the resultant zygote has 47 chromosome. Instead of 46, it has 47 chromosome. That is called numerical change in chromosome. I think you are understanding. I will give you diagram. Numerical change in chromosome is of two types, euploidy and aneuploidy. Euploidy is a duplication, diploid, triploid, tetraploid, pentaploid. And aneuploidy means one chromosome less, 2n minus 1, 2n plus 2, 2n minus 1, 2n plus 2. It means 46 minus 1, 46 minus 2, 46 plus 1, 46 plus 2. It may be the organism may have 47 chromosome, 48 chromosome, 45 chromosome, 44 chromosome. That is numerical changes in chromosome, and that causes genetic problem, genetic diseases. Then next topic is artificial induction, causes of mutation. You know, causes of the mutations are chemical mutagen. Chemical mutagens are chemical drugs, methylating agents, acridine dyes, chemicals. Then physical mutagens, they are radiation, solar radiation, X-ray, alpha, beta, gamma rays, high temperature. These are the causes of mutation or change in chromosome, sudden change in chromosome. Today we studied at mutagens, means artificial methods of mutation, numerical change in chromosome, polyploidy. Polyploidy is of two types, euploidy and aneuploidy. And then we studied about chromosomal mutation, deletion, duplication, translocation. You study it, we will see next day, next topic. Thank you.